Hello, IB Bio students. This is your next installment of Notes with Ms. Sheely. This particular notes is on enzymes, and this is section 2.5 in your textbook. All right, what is an enzyme? So we're going to review enzyme substrates and active sites. So we have our substrate here. This is the reactant in a reaction. We have our enzyme, which is a globular protein which acts as a catalyst for the reaction. Within the enzyme and the substrate, we have polar regions of amino acids, and that is what attracts the substrate to the active site and not to another section of the enzyme. When they are together, the substrate fits into the active site. So this is the region on the surface of the enzyme to which the substrates bind and which catalyzes the reaction. This then is called or referred to an enzyme substrate complex. So then the reaction happens. Once the reaction happens, it can be released, and the products are then released. And the enzyme, you can see, is the exact same as when it started, so it can be used over and over and over again. So we're going to look at what is an enzyme.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
So enzymes are specific to their substrates. This is what we've taught you in previous biology courses. There's the lock and key hypothesis, so that the substrate and the active site match each other in two ways. They must structurally match. The structure of the active site is specific to the substrate. If the structure or the shape does not fit, then they won't react. We've also talked about a chemical reaction. Substrates that are not chemically attracted to the active site won't be able to react. So if there's a positive or a negative charge that is not appropriately associated, then they won't react. But that's not 100% true. There's actually a better model. It's called the induced fit model, and this helps to explain enzyme activity a little bit more realistically. It is actually very similar to what you just saw in the video. So if the lock and key model were true, only one enzyme would catalyze only one reaction. And in actuality, there are some enzymes that can catalyze multiple reactions or work with multiple substrates. So as a substrate approaches the enzyme, it induces a conformational change in the active site. It changes to fit the substrate. So the active site changes, and you can see here we went from this nice sharp crisp arrow to kind of a much softer curvy arrow. That is our conformational change. But this change also stresses the substrate, and that is what really reduces the activation energy of the reaction. So that is what allows the reaction to take place, and then once it's taken place, the products will leave and the enzyme returns to its original shape. All right, just look at a quick little animation. So in this particular example, it's wrapping around. All right, so we just talked about the fact that that stress on the molecule in the enzyme is what lowers the activation energy of a reaction. But what is activation energy? It's the amount of energy that must be put into the reaction to make it happen. So here we have the red line being the uncatalyzed reaction and then the blue line being the catalyzed. So they're both going to require some activation energy to be able to get down to the products. So we're going to add some energy, and he gets up a little bit more. Add a little bit more energy, a little bit more, and look at how, as we're adding that energy, we're starting to stress the object or the substrate. More stress, more energy, more stress, all the way at the top. So this becomes the transition state. This is the full amount of activation energy that is necessary. So now the reaction can start to occur. Boom. And then the difference between our starting energy and our ending energy is the net energy change. So as the enzyme stresses the bonds in the substrates, it reduces the activation energy that's required for the reaction to occur so it can happen faster. All right, denaturation. We talked about this in freshman bio as well. Enzymes are globular proteins. Their structure can be altered by changes in pH or temperature. And if the shape of the active site is changed considerably, then they won't function. Denaturation is changing the structure of a protein, specifically an enzyme, so that it cannot carry out its function. There are several things that can alter this, mainly high temperatures, because the extra energy leads to increased vibration and it breaks the intermolecular bonds, typically holding the alpha helix or beta pleated sheet together. Also, changes in pH will cause denaturation because the hydrogen bonds are broken. Both of these are going to result in a completely different molecule and are typically irreversible. Let's look at a little video on protein denaturation.
This familiar gelatin dessert actually is a good example of the process of coagulation of proteins into a three-dimensional latticework that entraps water molecules to produce a semi-solid gel. This animation explains the physical chemistry behind the process. Proteins are synthesized by polymerizing amino acids. The polymerization occurs by repeatedly forming peptide bonds that link individual amino acids together into a chain. There are three structural features that influence the three-dimensional shape of a water-soluble protein. Primary structure is the peptide bond between individual amino acids that creates a long chain of connected amino acids. These long chains of polymerized amino acids have hydrophobic, water-repelling, and hydrophilic, water-attracting projections that are oriented perpendicular to the chain, as shown in this illustration of a growing protein chain. Secondary structure is the helix that the protein chain curls into as a result of hydrogen bonds and other weak forces. Tertiary structure is created when the protein molecules fold back on themselves outside of the helical segments to put the hydrophobic portions to the interior and the hydrophilic portions to the exterior. Several helical regions can exist in different portions of the molecule. When the protein has folded and refolded to reach its most stable configuration, it will have mostly hydrophilic amino acid residues on the exterior and mostly hydrophobic residues directed into the interior. When natural proteins are subjected to physical or chemical treatment, their structure changes and they become unnative or unnatural. We call that process denaturation. In this example, heating the proteins in solution imparts energy to the protein molecules. This added energy is enough to break the relatively weak forces that hold the protein in its refolded and helical tertiary and secondary configurations. As the process of denaturation proceeds, the protein molecule unfolds more and more, and the internally directed hydrophobic regions now become exposed on the outside of the molecule. The peptide bonds are largely hydrophilic. Once these segments are set free from each other, they attract water molecules. The recruitment of water molecules entraps the water molecules in close proximity to the protein strands. The hydrophobic portions of the molecules are also exposed. This situation is unfavored because the hydrophobic portions of molecules are not stable in an aqueous environment. Hence, upon unfolding, the hydrophobic regions on individual protein molecules will associate with hydrophobic regions on other protein molecules. This situation encourages the association of these protein molecules into larger and larger random three-dimensional structures. The molecules aggregate into very large water-insoluble collections that are quite randomly assembled. As the proteins denature, latticework structures grow amorphously and attract the solvent water molecules into cell-like structures. The self-associated water molecules, in groups, adhere to the surface of hydrophilic regions of the protein, while hydrophobic regions of the protein dissolve into each other and provide the energy to retain the structure. As this process continues irreversibly, all of the protein molecules are recruited to this large insoluble mass in a randomly organized structural framework that contains entrapped water molecules. One example of the consequences of unfolding and reassociating protein molecules is coagulation of egg white. Frying an egg is no more complicated than denaturing the egg white protein. The assembly of irreversibly denatured protein molecules results in formation of a solid gel. The gel entraps water molecules inside the white into a semi-solid structure, which holds its shape under normal conditions. Other examples of denatured protein assembling into three-dimensional structures include the baking of yeast-risen bread, coagulation of meat proteins by cooking in such products as hot dogs, and in the solidification of gelatin upon cooling of a solution. Denaturation and coagulation of proteins is a complex, irreversible process, but the study of denaturation has allowed us to better understand the three-dimensional structure of native proteins.
All right, great video on denaturation. All right, so there's lots of different factors that affect enzyme activity. Some of the big ones that we're going to talk about in class are temperature, pH, and substrate concentration. This is a really great little site. Take you there real quick. You can play with the number of enzymes that are available, how many substrates you have, if you have any inhibitors, what your temperature will be. And then you just kind of start and you see what's happening, how many products you have. So you have red products. What would happen if you added more enzymes? So what would happen if you took the temperature way up? So this is really neat. You can kind of play it around. See, I'm not getting any products. Why would I not be getting any products? So things to think about. We are going to do a lab where we kind of look at some enzyme activity as well. Let's look at the effective temperature on enzyme activity. So as temperature increases, the rate of the reaction increases as well. Because the molecules have more energy, they're going to move faster, and therefore they collide and react more frequently. But there is always an optimal temperature. If we go above this optimal temperature and continue to increase the temperature, then we are going to lead to denaturation of the enzyme. And the active site is changed, and so we have no function. Here we have a thermophile and a human enzyme. And thermophiles are bacteria at deep sea vents. And they're able to withstand a much higher, their optimal temperature is much higher on the range. When we're looking at pH and enzyme activity, there's also an optimal pH. Enzymes only operate within a very narrow range of pH values. So if we look at these two digestive enzymes, we have pepsin, which is found in our stomach, so it is a very low pH that it works in. And then we have salivary amylase in your mouth, so that has a much more neutral pH. So if there's a deviation from the optimal pH, you see there's a very steep line. So if there is a deviation, then the hydrogen bonds between the amino acids are broken. And then we have a loss of the shape, loss of the active site, and so we have no function. All right, the effect of substrate concentration on enzyme activity. We can increase the substrate concentration, and that will increase the rate of the reaction. But at a optimal point, or at a maximum point, all the active sites are full. It doesn't matter if you add another 100, 200, or 1,000 substrates. Your enzymes are working as fast as they can. So any increase beyond this will have no effect, but there is no damage to the enzymes either. All right, so lactose intolerance. Lactose is our milk sugar that's made of galactose and glucose, and lactose can actually cause allergies in some people. This is often because they're unable to produce the enzyme lactase in sufficient quantities. All right, so kind of interestingly enough, most people produce less lactase as they get older. After all, we don't live off of milk once we've been weaned. So we need to be lactose tolerant when we are young children, but then as we stop that need from mother's milk, then we start to produce less lactase. And there's some regions, such as Europe, there has been a mutation that has allowed lactase production to continue into adulthood. So this mutation is not present in people who are lactose intolerant. And you can see here we have a lot of lactose intolerance here in the red and the brown areas. So how can we cope with lactose intolerance? There's actually a couple different ways. One, we can take a supplement. So a supplement is produced by industry, and it uses a fungus to make enzymes. So it would make the lactase enzyme, and you would just take a supplement of lactase made by this fungus. You can drink lactose-free milk. Now, you have a couple different ways of drinking, having this milk be lactose-free. One is you have milk that has been given lactase, and essentially the lactase, also produced by the fungus, pre-digests your lactose before it's packaged. So before it's packaged, the lactase is added, then the lactase is able to break the bonds between the glucose and the galactose. But there's another way. Besides adding the lactase to the milk, the problem with this is that it is very low quality milk, and it's very wasteful of the lactase. There's no way to get it back. You're just drinking lactase. The other way is to run milk through an apparatus. So here is your apparatus. You cycle the milk through these tubes and through this tube, and in the tube are these little beads. They're called alginate beads. And on the alginate bead, you have immobilized lactase. So each bead can hold 2, 4, hold some enzymes. And then you cycle the milk through, and the lactose will bind to the lactase enzyme and will be split into the glucose and the galactose. And the glucose and galactose come out in the milk product. So it's lactose free. But the enzymes are still on the beads. They don't get washed away. So this is less wasteful of our lactase. All right, and that's it for our enzymes notes. Hope you enjoyed today. Come to class with questions.